Here's a surprising fact. Did you know that only 30% of the pregnancy end up with a baby? Most of the conceptions fail in the first month, and specifically, many of them fail during implantation. This is the stage in which the early embryo is implanted in the uterus wall. Failed implantation is a major limiting factor also in assisted reproduction, and yet it is very hardly investigated because its investigation is very hard in mouse, and in human is nearly impossible. And the reason is that while pre-implantation embryonic development can be recapitulated in the lab as done routinely in IVF treatments, post-implantation embryonic development happens inside the uterus and cannot be recapitulated in the lab. While in mice, we can take out embryos at different stages as much as we want, in human, naturally, we don't do that. And we very rarely have access to these early stages of implantation and post-implantation in which the structure of the embryo is starting to form. So early post-implantation development in human is a black box. That's why researchers in stem cell field were thinking to build a model for early embryos. And they were inspired by these beautiful structures. These are 3D structures which mimic an organ in a small scale, and they are generated from cells in the dish, in culture. So if brain and gut and retina models are possible, then why not embryos? And that's exactly what they did in the last few years. There were embryo models generated from cells of both human and mouse early embryos. However, growing them beyond implantation was impossible. And the reason that attempts to transfer them to surrogate mouse mother failed, and there was no method to grow them outside the uterus. That's where our lab enters the story. This is a roller culture. It was developed already in the 70s to grow embryos outside the uterus, but it was not very successful. And we thought maybe we can take it and improve it to grow embryos for a longer period. So we added a module which regulates the gas blend and pressure. And inside the bottles, we supply nutrients to the embryo so they can grow floating inside. The main role of the roller culture is to allow the circulation of the nutrients, which in this stage, they get to the different tissues by diffusion. This way, we took out natural embryos, mouse embryos, very early, at day 6.5 and even earlier, and grew them outside the uterus for 5 days. Why do we use mouse models? Well, we can do this in human, and mouse is a great animal model. It is very easy to grow, and its pregnancy is very short, only 20 days. So this stage is equivalent more or less to 6 to 7 weeks in human embryonic development. You can see here the movement of the embryo, the beating heart, and even the fetal blood inside the yolk sac. After day 11, the embryo is too big to get the nutrients by diffusion, and it starts to decay. And what we learned from that is that at least up to day 11, the mouse embryo is self-organized, meaning that it does not need the maternal tissue in order to develop. What can we do with this system? Well, we can do now live capture imaging of this embryo, for example, like the neural tube closure and the gastrulation embryo of both wild type and mutant. We can learn the dynamics and the mechanics of the embryos as they grow. And remember this? So can we now grow embryo models outside the uterus? That's exactly what we try to do. And we started with mouse. We took mouse stem cells, but when we just take stem cells and aggregate them, what we get is a mess. So we wanted to mimic the 3 cell types which reside in the early embryo of the mouse, and we did so by genetic manipulation, by inducing specific genes, and we generated these 3 cell types. And we continue to grow them in shaker and then in the roller culture. And after 4 days, we see that they spontaneously arrange into a 3D structure which resembles a 5.5-day embryo. This structure has an epiblast, which is the part that we later develop into the embryo itself. And it also has an extra embryonic compartment, which we later develop into the placenta and the amniotic sac and so on. We continue to grow these structures, and we see that the yolk sac is forming. And inside the yolk sac, after a few more days, we see a mouse model with neural folds and somites and tail bud and amniotic sac. So all these different substructures that we expect to see in an 8.5-day-old embryo. Yeah, we call them SEM for stem cell-derived embryo models. The SEMs are not identical to natural mouse embryos, but they are highly similar, as can be seen here with the stainings. So they express the right genes in the right place and time. And we could identify different structures, for example, these that will develop later into the brain and the tail and the bones and the muscle cells. And we can even identify cells which are primordial germ cells that will later develop into the eggs and the sperm. We can even isolate them. Just think, who are the parents of the pups that are going to be born from these species? Never mind. What we saw here is that we can take mouse stem cells and generate advanced post-implantation embryo model, which was quite amazing. Now, the next natural question was whether such models possible to generate from human cells. This is the really interesting question, and that's what we tried to address. And at the beginning, we used a similar strategy to what we used in mouse. We took human stem cells, and I will spare you the details, but at the end, we found out that we don't need to do genetic manipulation to generate the 4 different cell types that we need to aggregate, but different signaling molecules are enough to differentiate and generate these cell types. So we generated these 4 cell types, we aggregated them and grew them, and after a few days, we see that spheres of trophoblasts, these are placental stem cells, are starting to emerge, and in less than 3% of them, there is a 3D organized structure. And we continue to grow them for 2 more days, and we see structures which highly resemble a 14 days human embryo model. Human embryo. These embryo models have epiblast compartment, which will, as I said, it will later generate the embryo itself. The epiblast grows, it cavitates. It also breaks symmetry, meaning that it starts to form the axis of the embryo. They have extra embryonic compartment, including a primary and secondary yolk sac, and they are covered by trophoblasts that will later generate the placenta. These trophoblasts start to differentiate, and they even secrete the pregnancy hormone, which we can identify by a standard pregnancy test. How cool is that? So the protocol is not perfect. Its efficiency is still very low, less than 1%. But for us, it's a proof of concept for our ability to generate an embryo model of implantation stage from human stem cells. And we are starting to open this black box. Once we are able to increase the efficiency, there is a big potential here. We can do things like use it as a global differentiation protocol to produce cells that we cannot generate in the lab so far. We can study developmental defects, which are very prevalent in this stage of embryonic development, and we can do drug screening to test drugs which, if they affect these embryos in such an early stage. So to sum up, what we saw today is that mouse embryo is self-organized, at least up to day 11, so it does not need the maternal tissue in order to develop. We saw that we can take mouse stem cells and generate the 8.5 days embryo model from them, and we can do the same with a human stem cell generating a 14 days model, which is a very important window to implantation. With this, I would like to end and thank the amazing students in Jacob Hana's lab that did all this work, our dear collaborators, the funding agency, and thank you for listening.